It's my pleasure to begin by welcoming everybody to the very first Virimaris webinar. My name's Ian Carr and I'm responsible for business development at Virimaris, and I'm really very happy to be able to connect with you all today via this webinar platform. Our purpose today is very simple. We want to share our passion for healthy fish, and we want to discuss the role that Optimum Omega nutrition plays in helping farmers to achieve that goal. And whilst the topic is relevant today for aquaculture producers in broad terms all over the world, no matter what species you're farming, our focus today is on salmon farming, and with a small bias as well towards Chile for this particular webinar. So in terms of process, the webinar today will run for an hour overall. The session runs in English, but we have a good number of Spanish-speaking people joining today, so we have a live translation into Spanish too. Please follow the link when you log in to launch the simultaneous translation. First of all, we're going to hear a truly expert overview of the subject area from Professor Brett Glencross, who we're delighted to have with us here on the webinar today. And that will give us substance then to discuss further with our panel, which will include Felipe Almendras, the Director of Green Volution, and our very own Jorge Torres, Business Development Lead for Virimaris in Latam. And Felipe and Jorge a little later on. You can also take part in the discussion today by registering questions directly in the webinar portal. You'll see the panel. It's on the right-hand side of my screen, hopefully the same on yours, and you'll see an area there for questions. Please register your questions and we'll get to as many of those as we can in the time that we have after the presentation. But as an outcome for your time invested today, we want to show you why we think algal oil as a source of EPA, DHA and ARA helps farmers to take care of the health and therefore also the welfare of their fish by applying Optimum Omega nutrition as a tool. And we'll explain how we think the benefit of this plays out in terms of higher levels of productivity for salmon farming operations as well. So let's start then by learning a little bit more about why EPA, DHA and ARA levels matter through the production life cycle of farmed salmon. And let me briefly introduce you to Brett Glencross. Brett is currently the Professor of Nutrition at the Institute of Aquaculture at the University of Stirling here in Scotland. Over the past 25 years, he's worked in various academic, institutional and industrial roles all over the world, and therefore he's well known to many of us through his close work with farms and feed companies throughout Australasia, the Middle East and also Europe. And Brett's going to give us an overview of his very many academic achievements at the beginning of his talk. So without further delay, Brett, I'll hand over to you, and we are very much looking forward to your talk.
Thanks for that introduction, Ian. So with our talk today, what we're really going to focus on is the metabolism, function, and requirements for principally EPA and DHA in Atlantic salmon. But we'll also basically maybe segue in some cases into looking at the roles of other fatty acids like arachidonic acid, as Ian mentioned. My background, I started doing research in aquaculture about 25 years ago, and my PhD was initially on this very topic and actually understanding the requirements for essential fatty acids in shrimp. And then about 10, 15 years ago, I started doing similar work, understanding the requirements for essential fatty acids in Asian sea bass, or the barramundi, which is a marine fish. And then on moving to Scotland about 5 or 6 years ago, I started working on Atlantic salmon, and we started doing a lot of work there, really building on the great work that the Stirling Institute have been doing for the last 30 years in lipids area, and getting more detailed now into understanding the intricacies of the requirements for certain long chain fatty acids in the diets of species. So a lot of my career has really been spent exploring the nutritional demand for essential fatty acids by aquaculture species, which ironically was the title of a review that I wrote about 10 years ago. Now, when we talk about essential fatty acids, there's generally 6 that we're referring to. 3 of those are omega-3s, 2 of them are omega-6s. Omega-3s tend to be short chain linolenic acid, or the long chain icosapentaenoic, EPA, or docosahexaenoic acids, DHA. Whereas the omega-6 is 1 short chain and 1 longer chain ones. Now, just as a quick reminder, the 18 refers to the number of carbons in the chain. The 3 is the number of unsaturated bonds. The N3, or the omega-3, tells us where the first bond is in relation to that end of that fatty acid chain. So 24N6, 20 carbons, 4 unsaturated bonds, and the first one is 6 from the end of that chain. Right, so what really defines biologically this issue of essentiality? What defines the need for a nutrient in the diet on an essential basis? Well, a lot of it's to do about how well an animal can actually make a nutrient. So all organisms, plants and bacteria and animals, can make 18,1N9, oleic acid. But the conversion of this 18,1 to 18,2 or the 18,1 to 18,3, only plants can do this. And principally algae are great at it, so are some terrestrial plants. Once the polyanids are formed here, the 18,3 and 18,6, there's a wide variety of organisms that can then change these fatty acids into the more unsaturated and longer chain varieties. But not all animals can do that. In fact, this was what defines whether an animal needs shortened polyunsaturates in its diet or whether it actually needs to be provided with the long chains preformed. In fact, usually what it is, it's the absence of this enzyme here, this delta 5 FADS1, which is often what demarcates those that need to be given preformed long chain, or whether they can actually take the short chain and make some of the long chain that they need. Another important aspect is that the machinery that does that conversion, it varies depending upon what tissue you look at. This is an example of a study that was looking at tissue from the brain, liver, kidney, and the gill in Atlantic salmon. And if we just focus on those 3 key omega-3 enzymes of the 5-FAD, the 6-FAD, and the ELA-VL5, amongst the different tissues, we can see that the expression of the genes for those enzymes is quite variable depending upon the tissue. For example, in the liver, we can see that the 5-FAD is highly expressed, but it's basically not expressed in the kidney at all. And this difference really reflects the metabolic demand by these tissues for these long-chain fatty acids. So for example, liver and brain need long-chain omega-3, but the head, kidney, yeah, not so much interested in it. But what about on a whole animal basis? I mean, Atlantic salmon is quite an interesting animal in that it has some ability to make these long-chain omega-3s itself. So in this study, which was done only in the last year or two, it examined the feeding of first feed and fry, where they were fed diets that basically had no long-chain polyunsaturates at all in the feed. What they were provided with was the short-chain polyunsaturates of 18,3 and 18,2, and the ratio which those two omega-3 and the omega-6s were included in the diet was changed at a ratio of 3:1, 1:1, or 1:3. Now, the fish that were fed those diets, they grew fine. They seemed apparently healthy. But when they compared them to the growth of the animals that were fed preformed long-chain polyunsaturated fatty acids, the growth was much poorer. So what it clearly showed is that these animals can make this stuff, but they would really much like it actually pre-supplied in the diet in a preformed manner. What you can sort of see here, this is showing you the production that was achieved of the omega-3 in the animal. And what you can sort of see is that the greater the amount of the 18,3 that was provided to the animal in their diet, they made more of the EPA, DHA, and DHA in their own bodies. In fact, importantly, if we look at that level of the ratios of DHA to EPA that was present in the bodies of the animal when the animal was forced to make it itself, the ratio varied from 3:1 to 5:1. So when the precursor, the 18,3, was highly abundant, the salmon made it at a rate, DHA to EPA at a ratio of 3:1. And when the precursor was not abundant, it made it at a ratio of 5:1. So you can sort of see the animal is actually targeting in its own machinery to make a ratio of DHA and EPA in its body. So what do animals do with these fatty acids? Well, they actually play a variety of roles. DHA in particular plays a very important role in cellular development, cellular synthesis. It plays an important role particularly in things like brain cell development, nerve cell development, and eye cell development. It's principally playing a structural role there about maintaining membrane fluidity of those cells. EPA and ARA, on the other hand, they play a slightly different role. These are what we call precursors to making hormones. In fact, in particular, the arachidonic acid, the omega-6, is actually the main precursor to these series of enzymes, the prostaglandins and the leukotrienes. Now, these hormones play a range of roles, but in particular, they play an important role in chemokine production, immune cell activation, and inflammation. Now, arachidonic acid, as I said, was the main precursor to making these hormones to actually regulate these processes, but it's the go signal. It actually has a positive inflammatory response in terms of its role. To counter that positive inflammation response, EPA comes in. EPA has an anti-inflammation response. So it's sort of like the yin and yang in terms of the keeping the balance of that inflammation response. You want inflammation to actually help resolve tissue damage, but then you need to actually take inflammation away. And that's where EPA comes into its role. So let's look across these three important essential fatty acids and look at a bit of a summary of what their different roles are. Each of them plays an important role in reinforcing the immune system of animals, particularly salmon. EPA plays a really important role in enhancing the pathogen response and survival of fish. All three fatty acids play a role in smoltification, although DHA and ARA seem to have more role in the freshwater stage, whereas EPA's role seems to be more into the saltwater stage post-transfer. All three play a role in skin integrity, robustness, and wound healing, whereas ARA and EPA seem to play a role in stress regulation, probably linked to their roles in the prostaglandins and leukotriene hormones. All three play a role in effective cell metabolism and energetics, and DHA and EPA in particular play a role in bone mineralization. So they contribute to a range of cellular function and tissue function in the animal to maintain health and robustness. So that's the story about what they need. That's a qualitative analysis of the three key long-chain fatty acids. What about a quantitative analysis? Well, if we go back to the original work that was done about 20 years ago by Bente Reuter at NOFEMA, what we can sort of see here is as we increase the supply of different fatty acids in the diet, there was no response to changing of the short-chain omega-6. Introducing the short-chain omega-3 resulted in the best response at an inclusion of about 1% of the diet. But this was eclipsed by the performance that we got when we included around about 1% of the long-chain polyunsaturates in the diet. But an important element of this is that the best response we saw to growth was the best response we saw to mortality. In fact, the lowest mortality was seen at much higher levels, between 1.5% to 2% of the long-chain polyunsaturates in the diet. More recently, people have started to do a few more studies in understanding these quantitative responses to DHA and EPA. There's a series of two studies here, one by Glencross in 2015, and another by Marta Bao from NOFEMA in 2017. And both studies were very consistent with each other. They largely reported similar findings in identifying that the optimal requirement for growth, at least, appeared to be between 10 and 15 grams per kilogram of diet. That's about 1 to 1.5% of the diet. What it also showed from this work is that it didn't seem to really matter whether the fatty acids were provided as EPA and DHA, EPA only, or DHA only. The animal seemed to be relatively ambivalent as to that omega-3 and how it was provided, obviously because it has that machinery and the ability to transform things into the actual molecules it needs to a certain extent. More recently, there was another study published by Greta Rosalund from Skretting that looked at the similar sort of style study, but from a much longer term basis. In this case, now looking at almost the full production cycle of salmon. In this study data here, what we're looking at is the retention of the EPA and DHA of the fish, and we're looking at it at two different temperatures. Now, what we can see from this data is that the retention basically plateaus between 3.4 and about 5.2% of the diet. That's equivalent to around about 11 to 15 grams per kilogram of those fatty acids in the diet, similar to what we saw from the data from Glencross and Bow. So that sort of really consolidates that information there. What it also shows is that the retention or the utilization wasn't affected by temperature. So we don't necessarily need to change our inclusion of these fatty acids, certainly within normal operating temperatures, in terms of what we need to provide for the salmon in the diet. The other interesting thing about this study was that the authors actually presented the fatty acids in terms of the percent of fatty acids in the diet, not as a grams per kilogram of the diet, but as a percent of the total fatty acids. And that raises another important question, is how do we actually express our requirements for fatty acids? So this was a study that was published this year that actually answers that very question. What it looks at is a study that has two different lipid levels, high lipid and low lipid. So high lipid is around about 23%, low lipid was 80%. We're dealing with small fish here, so obviously the diets are relatively low in lipid. And two levels of omega-3, a low level of omega-3, about 7 grams per kilogram of the diet, and 15 grams per kilogram of diet in the higher omega-3 level. And the fish were fed for around about 3 or 4 months. And this was the responses that we got. And you can sort of see a very nice pattern here associated with the way that they're presenting those results. Now, if you transfer that data across this figure on the right here, those treatments pretty much correlates to about 3%, 4%, 6%, 8% of total fatty acids in the way in which they were representing there. So we've got two lipid levels and the two fatty acid levels, allowing us to check that factorial design. What you can sort of see is in the high lipid diet, there was a clear difference associated with the inclusion levels of fatty acids, but in the low lipid diet, we didn't see that response. It's what we call a clear interaction effect. Now, what's the implications of this? Well, quite clearly, what it means is that the fatty acid requirements for an animal change as a function of lipid level in the diet. So if we take those 4 treatments from the previous experiment and overlay them on this panel here, A, B, C, D, what you can sort of see now is how they actually explain the performance of the animals with respect to the response to the fatty acids being supplied on a total fatty acid basis. So if I was to look across this figure here and take a transect, this would be sort of the response I'd be seeing across these colors. Obviously the red is the stop signal, we don't want to go down there. Amber is caution, green is where we want to be. Green is where we want to basically be including the omega-3 in our diets for Atlantic salmon. Moving on, so what about the supply of DHA versus EPA? We saw some of the earlier studies that the animals didn't seem to be influenced in terms of their growth, whether the fatty acids were supplied as DHA, EPA, or a combination. But that's for growth. What about health? Well, this was a study published around about almost 10 years ago now, came out from some work led by EWOS, where they actually looked at that question by varying the DHA to EPA ratio. In this case, you can see what they've done is they've increased the supply of EPA relative to DHA to decrease that DHA to EPA ratio. And they've fed it to salmon for 12 weeks, and then they've challenged those salmon with reovirus. Now, what they've then looked at is the viral load in the heart tissue of those fish, and what we can clearly see is the lower DHA to EPA ratio has clearly helped the animal better mitigate or manage the viral load in its heart tissue. Also, when we looked at the average histoscores, which is, I guess, the way of looking at the inflammatory response in those tissues, that lower DHA to EPA ratio, principally driven by the higher EPA level, also means they had basically a lower histoscore, which means they had less inflammation. The EPA is helping the animal regulate that inflammatory response better. Some other interesting aspects that are coming out, particularly in recent times with regards to the roles of these different fatty acids, and particularly the balance between EPA and ARA, was just some work coming out of IMR in Norway, where they were looking at basically a study where they had the EPA, DHA, and ARA kept constant in diets. But what they then is they varied the presence of that short chain omega-6, 18-2. And in that case, what they can see is that as they increased the level of 18-2 in the diets, what changed was the level of arachidonic acid, EPA, in the total lipid, in the total fatty acids of the polar lipids, the structural lipids in the liver. So what was happening was that the presence of this short chain omega-6 was clearly promoting the production of arachidonic acid in the liver, where it was actually also then down-regulating or regressing the levels of EPA in the liver. And this, of course, has implications in terms of the animal's ability to regulate its inflammatory response. When we look at the role of linoleic acid, this 18-2 in different tissues too, as we increase the supply of 18-2 in the diet, certain tissues are very tightly regulated. The animal's trying to control how much of the 18.2 goes into those tissues, because it wants to keep it down to actually try and avoid that inflammatory response. Things like the eye, the retina, the blood, the heart. It's trying to regulate and control that fatty acid profile. Other tissues, for example, like the muscle, it's not so concerned about that. And what you can see there is that as we increase the 18.2, it's pretty much coming directly reflected in the muscle, because it's muscle lipids are largely just generally a storage reflecting off the diet. Similar bit of work that was done about 20 or 30 years ago by Gordon Bell, one of my colleagues from the Sterling Institute. He's retired now, unfortunately. They looked at the inclusion level of sunflower, which had a very high levels of this 18.2 in the feed. And what they noticed when they actually looked at the presence of cardiac necrosis in salmon fed these diets was that high levels of that linoleic acid, the 18.2, really exacerbated the number of fish that had cardiac necrosis, whereas high levels of fish oil seemed to result in normal heart development. So what we're seeing here is two clear dichotomies in terms of the roles that omega-3 and omega-6 play in terms of their roles in inflammation. The two key fatty acids there in that inflammation regulation process are EPA and the ARA. And this has also been found in a lot of studies with human work, where they see that EPA in particular plays a really strong positive role as an anti-inflammation mediator. Now, DHA can play a role. It does also go to some of these roles in productions of these hormones as well, and it can also be retroconverted to make deep EPA. But the problem is that all retroconversion comes at a cost. It's arguably better to actually supply the substrate in a preformed manner rather than making the animal continually make it at its cost. The other key thing about the supply of omega-3 is one of the reasons we feed it to the animals is not just for their needs, it's also for our needs. We're looking at salmon as a principal omega-3 source in our diet. And we can sort of see that the effects of fatty acids on the animal's diet has a direct impact on the fatty acids we see in the whole body of the animal. We can also see that that directly also transfers in terms of what the composition is of the fatty acid profile in the meat, because there's an incredibly strong relationship between the whole body fatty acids and the fatty acid composition of the meat in salmon. Now, why that's important, or at least interesting, is that that feature really lends itself well to our ability to model what happens to fatty acids when we start looking at the application of different diets. So for example, if we model the growth of, say, a small fish becoming a big fish, and there's a range of different growth models there we can apply that information to, that growth model, of course, dictates feed intake. And of course, the feed intake then we can then design based upon what fatty acid profile we have in that feed to then influence the deposition efficiencies of those fatty acids going into the whole animal. We can then also under the composition of the animals at time point A and time 2 and model the whole body of the animal, which of course then links to the composition of the fatty acids and lipids in the fillet, which we can then predict what the composition of that NQC, that fillet portion, is going to be. This is actually really quite a predictable process, and at the moment we're working very closely with Veramaris to actually lead in the developing of such a predictive model for application into the future. Why is this also important? Well, one of the problems we've seen in the last 10 to 20 years is there's a race for the bottom. Obviously, with constraints to limits of fish oil supply around the world, feed industry has basically had to start replacing fish oil with alternative forms of oil. And the two main oils they've looked to use is really rapeseed oil and poultry oil, both of which are high in non-long chain omega-3 fatty acids. So fatty acids like 18.1 omega-9 oleic acid, 18.2 omega-6 linoleic acid, and 18.3 omega-3 linolenic acid tend to be these fatty acids that come in with plant oils. The long chain omega-3 like DHA and EPA tend to be the ones we associate with fish oils. And so what we can sort of see here is that as we've progressively begun replacing the use of fish oils over time, is we've now arrived in a position where the level of omega-3 in salmon, farm salmon, is arguably less than half of what it was 10 to 20 years ago. And the levels of omega-6 and omega-9 are now at least double what they used to be 10 to 20 years ago. So what's the implications of that in terms of quality and the level of DHA and EPA and fillet quality in salmon? Well, some recent work coming out of Nofima in Norway, they looked at the application of a microalgal source of DHA through the use of a schizachytrium biomass, and they applied it to diet and then compared it against fish oil. Now, interestingly, they actually found that the use of the schizachytrium oil actually outgrew the fish fed the fish oil. But they also had other positive features too. They had high levels of EPA and DHA in the fillet, better pigmentation in the fillet, and also a reduction in melanin spots. And they also found through sensory analysis that there was improvements in color of the meat as well. So where does this leave us in terms of a summary of how we look at understanding the requirements for EPA and DHA in Atlantic salmon? Well, below a critical threshold, there's clear deficiency, generally around about, say, 1% of the diet or less than, say, 2.5% of fatty acids in the diet, we run into problems with performance. There then becomes a realm basically where normal conditions under, operating temperatures which are within the normal boundaries, unstressed fish growing fast without any problems, they generally grow well, somewhere between 1 to 2% long-chain polyunsaturated fatty acids in the diet, which equates to around about arguably 3 to about 5% of the total fatty acids in the diet. Now, when we get the challenging conditions, because we all know that no one farms under normal conditions, what we see is that the leading farmers and scientists are now actually suggesting that we need to perhaps reverse that trend we've seen in the last 10 to 20 years, where we've bled the fatty acids out and start going back up to increasing the levels again, simply because it actually helps with that robustness. In fact, what we need to look at is a new way of thinking. So in recent years, what we've probably seen is the Chilean industry operating their supply of their omega-3 levels somewhere in this sort of range, somewhere between the 1% and 2% of the diet, which equates certainly in the smaller fish feeds, the lower fat feeds, in a range that's probably useful. But certainly when we get to the higher fat diets, where we're using the majority of the feed, we're now getting into the area which is, I guess, in the amber zone. Those industries or those growers who actually are leading, they seem to be taking a different approach. And they seem to be pushing their omega-3 back up towards these higher levels of total fatty acid supply, because this is what this new way of thinking. We need to think about fatty acids being supplied as a proportion of the diet. It's a relative requirement, not an absolute requirement. This is where we're seeing those present their feeds. So what we're seeing is there's a new way of thinking we need to look at with omega-3, and it's about increasing that supply of omega-3 as we increase the lipid in the diets for salmon. Thanks for your attention.
Okay, very good. Thank you very much, Brett, for that very interesting and thought-provoking talk. And I want to just start by recognizing you for your personal contribution to this whole body of research. It's difficult to go through the scientific contributions to omega-3 nutrition and omega-6 nutrition without seeing your name on a lot of the papers. So appreciate that, and also for the time you've taken to prepare for this talk today and guiding us through that so well. I think, as you say, we've got a new way of thinking here, and it seems to me that we're really approaching an interesting point, where the industry has been, in a way, economizing on omega-3 in particular, because it's had to over recent years, and yet now the scientific community is saying, well, actually, there's benefits to be gained here by increasing the overall omega nutrition, and also paying more attention to the types of fatty acids that we have in the diet and the balance of those throughout the life cycle. So do you think then we now, the industry has sufficient clarity from the scientific community in terms of how to do that? And where do you think the direction of current research is going at the moment in this whole area?
As I sort of probably alluded to in some of that presentation, a lot of this research, particularly the role of EPA versus DHA, this new way of thinking of fatty acid supply, this is work that's come out in the last 2 to 3 years. I mean, yeah, that paper of mine that we published about the relativity of fatty acids, that was published this year. So it's really new stuff. The scientific publishing process, from my experience, is not a great way of communicating with industry. You're much better off actually interfacing with them directly through collaboration or through meetings and conferences. Of course, that's a bit challenging in 2020 for a range of obvious reasons. But it's about basically also providing vehicles like what we're doing right now with this webinar to actually present that information in a cogent series of snapshots that actually show what the cumulative mass of weight of evidence is saying. We see things like, a lot of the research as it's sort of showing is that the animal didn't seem to worry about whether it was fed EPA DHA or EPA plus DHA in terms of growth. And a lot of the academic studies in the last 20 years were very growth centric. We're only now starting to revisit some of that in terms of understanding things like inflammation mechanisms and responses to disease challenges and the like. And that's changing our view in some respects. It probably puts us a bit more in alignment with the way industry is thinking in, right, how do I deal with feeding my fish under challenging conditions? There's one thing about what the animal needs when it's not challenged, but there's also that what the animal needs actually when you take it out of its comfort zone.
Very good point. Great. Okay, so we'll let you just take a breath, maybe grab a bit of water or something. And I just want to remind the audience, we've got a really good audience here today. Now's your opportunity to post questions in the webinar portal. There's an area for that in the portal. On my screen, it's in a panel on the right-hand side. You should just be able to enter your questions there, and we'll try to get to a few of those as we go through this panel discussion. But let's bring some more voices to the table then, to give Brett a little time to breathe, and we'll welcome our remaining members of the panel for today. If I could ask Felipe Almendras to join us. Felipe brings 25 years of experience in aquaculture and currently runs his own consultancy offering green solutions across the spectrum of additives, biologicals, genetics, and also consultancy. Felipe, welcome.
Thank you very much. Thanks for the invitation.
And then also, if I could ask Jorge Torres to join us as well. Jorge brings 20 years of experience to the table from his former roles, most recently as sales manager for DSM in Chile. And for the last 2 years, he's leading the business development effort for Veramaris in LATAM. Welcome, Jorge. You might be on mute there, Jorge. I don't hear you when you speak.
Yeah, sorry. Thank you for, and also thank you all the participants for joining us in this webinar.
Perfect. Okay, so then let's move to a few questions then for the panel here today first, and then we'll take a break and have a look and see what questions we've got coming in on the chat, and dance in a moment a little bit with those. But first of all, I want to just talk about survivability. And I think, Felipe, this is probably going to be a question for you with your background, your veterinary background. I recently saw the report from Kontali on their productivity barometer. And it really struck me how productivity of the salmon industry varies an awful lot between different countries. And there still seems to be a lot of room for improvement because the level of losses after transfer to sea still varies in the band of sort of 20 to 30% of individuals. So that means that there's a big number of individuals that are not making it through to harvest. And it's only the Faroes that consistently delivers levels below 10% across multiple generations. So what's your view on this? Why aren't those numbers coming down, and how can Optimum Omega Nutrition help in that regard?
Thank you for the question. It's very interesting. I've been following Bakkafrost like probably many of the salmon farmers in the world. It's a fantastic company, and they've been consistent in good numbers in that. One of the things that you check in their presentation, I heard a few presentations of them, and also in their annual reports, is that they in the strategy, they highlight in the core strategy the use of high omega-3 diets. That's something that I think is very interesting. It's not just about financial numbers, it's also about biology. And second is they also care a lot about marine ingredients inclusion. So they take care of these things forever, maybe because of their background. And there is one customer in Chile I know of that is also having exactly the same approach. I know they use 3% EPA DHA in the diet, and they have been consistent in showing that that brings them excellent results. The challenge of increasing the survivability in freshwater is big. We know that the number of eggs that the farmers use per small produce is quite high. Usually they account not only for survival in terms of sanitary issues, but also unexpected reasons, some other reasons that come up in the production in freshwater. But still it's a high use. It's about 70% loss that what they budget for in the freshwater process. It's still quite high. I was once with a farmer with usually they use, I don't know, 1.6 to 2 to 1 smolts eggs. That's usually the ratio where they start with. And it struck me too that there was once in Denmark, visited a large farmer there of trout, and their survivability was over 90. So I mean, those are numbers that you just don't see in Chile, no? So I think we just to make a long story short, what is relevant, I think, is that everyone is focused today in getting rid or decrease as much as possible antibiotics. So one of the things we need to focus on is what we put in the diet in order to make sure that we try to help the fish to do it naturally and deliver smalls antibiotic free, so to have a strategy of higher price and market positions.
Very good, fully understood. So then, Brett, assuming we can keep some more of these fish alive, the next thing we've got to worry about is their health and their welfare. And welfare in particular is an issue that is much more prominent on the agenda as it should be these days, because we know that fish are sentient animals. We know that they have memories, they can feel hunger, they can feel pain, and so on. So how does Optimum Omega nutrition contribute to fish welfare from your perspective?
Well, for me, it's principally about how the animal manages its disease and inflammation responses. I mean, if the animal is basically adequately equipped to deal with threats, inflammation threats to its well-being, then it actually means it's more resilient and robust to those challenges as they come along. But that's about actually making sure that they're not operating from a nutritionally compromised position. That's, to me, is about that balance of EPA and DHA and adequate levels of the omega-3 in the diet, such that when the time comes and they have to manage an inflammation response, or they have to make sure that their skin and their mucus levels are fully formed and robust, that they've got the materials to do it. It's about making sure that they have that resilience, that ability to deal with change when the needs come.
Okay, good, thank you. And Jorge, to bring you in then at this point, Felipe mentioned antibiotic resistance, and I'm just very well aware that conscious consumers these days are much more concerned, about the food that they're buying. They're much more aware, and many of them are looking for free from claims, including free from antibiotics. And I know in Chile there's been a project running called the PINCOI project, where stakeholders have worked together to address the use of antibiotics. So from your perspective, how do you think Optimum Omega Nutrition can contribute to that overall objective?
Yeah, thank you for the question. Yes, the consumer, you mentioned consumers are demanding feed free from antibiotics, not only for salmonids, for most of the protein they consume. And there is the PINCOI project plus the Chilean Salmon Antibiotic Reduction Program, which both are two examples of the challenges and the challenges condition that is facing the industry. And as I mentioned, not only salmonids, and also how collaboration and knowledge creation is key for the future of our industry, and also, in this case, an effort to reduce antibiotic consumption. So both programs have the same object, that are using an holistic approach where nutrition has a key role, and of course, Optimum Omega Nutrition is part of this solution, as was presented by Brett. And with the actual evidence presented today by Brett, and also more scientific information coming soon, we are sure that Optimum Omega Nutrition will increase the fish health, the fish welfare, will contribute to a more sustainable growth of the aquaculture, while transferring this benefit of higher level of EPA and DHA to salmon consumers.
Okay, very good. Thanks, Jorge. Then with that, let's have a little look here, see what questions we're getting in from the audience. And I see quite a few up here. So then that gives me the power to choose which ones we ask and which ones we don't. I think first of all, there's some questions about whether or not the content will be available after the webinar. And I know that Natalie is working away in the background recording everything, and that will be made available to people afterwards. So you can relax and you don't need to take notes too diligently. Then there's a question here on the pressure on fish oil supplies. And it's a great question. What are the different sources of EPA and DHA that we can use to increase the inclusion in the diets? Who wants to take that one?
Well, I can take that one, and perhaps Brett and Felipe can comment as well. Well, today Veramaris is the only source of non-marine, non-GMO natural source of EPA plus DHA, as Brett mentioned, plus arachidonic acid. So it has a very a fatty acid profile that fulfill what the fish needs. And if you see the omega-3 and omega-6 ratio is very similar to fish oil. So you can use Veramaris as a reliable, renewable, and reduce the dependency of forage fish on to grow the aquaculture in a sustainable way. So for sure, you can use our product in order to fulfill and increase the EPA DHA requirement from the fish, and also to decrease the dependency on forage fish.
Excellent. Anything to add to that, Brett?
Look, there's probably a range of things I can add to this. I mean, through various aspects of my career, I've been involved with some of the GMO work and some of the microalgal work and some of the fish oil work. And there's probably the three main areas where we can look at this long chain omega-3 supply story from. There's two GMO rapeseed products that are being commercialized at the moment. The Aquaterra from New Seed and the Latitude from BASF. Both of those tend to be, well, one's EPA centric, one's DHA centric. Total level of long chain omega-3 is only about 10%. There is another product that's in development with Rothamsted Research here in the UK, which is a much higher level of omega-3, but they're a little bit further away from commercialization yet. The microalgal story, mostly driven by, I guess, probably schizochaetrin production. It tends to be, for most parts, DHA centric. There is obviously some nanochloropsis production, which of course is EPA centric, but very few seem to be actually doing both. I mean, the Veramaris algal oil is probably the exception there. It's really quite a unique product in that regard. It's, from my way of looking at it, it's the only thing that really competes with fish oil properly in that true sense, and it's looking at that balance of the EPA DHA story. And the fish oil story, of course, in the fish oil story itself is a bit of a broad church as well. You know, the omega-3 levels in fish oils go as low as say 10% and as high as 30% if you get a really good quality, say, you know, Chilean or Peruvian anchoveta oil. And the DHA and EPA ratio can also vary markedly depending upon what oil you get. You might get a high EPA, lower DHA anchoveta oil, and that varies depending upon like where you go off the west coast of South America to get those oils too. So there's a bit of variation there in terms of those products. It comes out from my view, really understanding your materials and what it is that you actually want to put in the feed. And from my view, that's about the level of omega-3 and the balance.
Very good. And there is it's probably worth mentioning, there is a paper actually that was published by Professor Tocker and the team at Stirling last year, which gives a really nice overview of all the different possible sources and the pros and cons of each. So that's another paper that would be worth having a look at. But before, let's move on a little bit here because there are lots of questions. And one of them is, actually there are several that relate to other species, because this webinar today we're focusing on salmon. We did have plans to expand onto other species in future webinars, but maybe just briefly, Brett, you could just comment upon how Optimum Omega nutrition applies for marine fish and also for shrimp as well, very briefly.
Well, with marine fish, particularly things like sea brims and sea basses, they appear to be a bit more sensitive to perturbations, changes in the EPA DHA story than salmon. Salmon are relatively forgiving. They can still be temperamental, but when the fatty acid story you look at, sea brim and sea bass give you a much clearer yes, no, bad performance, good performance result. And shrimp also. Shrimp are also, because shrimp don't tolerate very high levels of lipid in their diet, they are also very quite responsive to changes in the long chain polyunsaturates in their diet as well. So they tend to be a little bit more sensitive in that respect. The data that we've got looking at changes of EPA DHA ratios, for example, things like gilt-edged sea brim, shows a lot greater sensitivity. They really do need that EPA as well. Whereas, as we saw with the growth work with Atlantic salmon, EPA, DHA combination, yeah, they'll grow okay. But sea bass and sea brim, no, you've got to have both there or you'll get problems.
Great, okay. So picking up again on some of these questions in here, there's a little bit about the balance of different fatty acids. So maybe let's just talk about EPA, Brett, for a minute, and just expand a little bit on your views on that. As I think as formulators start to get more sources of long chain omega-3s available to them, how much attention should they be paying on the provision of EPA in salmon diets? And why is that important?
I mean, one of my past roles in one of my former jobs, actually, I was a technical manager at a major feed company in Australia, and I was also a formulator for that company. So we would often look at how we would formulate diets, and the main thing we would be looking at often would be the total level of omega-3, not necessarily the breakdown with EPA and DHA. I think that's changing, particularly as we get a better understanding of the sensitivity of different species and the role that the fatty acids play, that we're not just formulating to a total omega-3 supply, but also the ratio of DHA to EPA is becoming important as well. I see this as becoming more and more important through time, particularly as we become more mindful about the health benefit and the welfare benefit and the issues associated with that, with the various species we're formulating for.
Very good. Okay, great. Now there's a few in here about fish quality, in particular melanosis. So I think, Felipe, that gives a nice platform for you to talk a little bit about that, because we know that incidence of melanin spots is a concern in the industry. It's been increasing in Chile, with some farmers I hear sort of mentioning even numbers as high as 25% prevalence in the harvested fish with melanin spots. Of course, that has a big impact on downgrades. So how much attention then should we be paying to fatty acid profile of the diet, and what role can Optimum Omega nutrition play in optimizing fish quality?
Okay, I think NOFIMA has shown in the last years, probably the last 5 years, that the increase of melanosis in Norway is dramatic. It has increased at least five times in the last 5 years. So that's pretty much coincidental with some studies showing that when you adjust diets in omega-3 and increase the levels, those melanosis at harvest decrease dramatically. So I'm not saying that this is the only cause. Yeah, it could be also associated to other things. For example, PRV is a virus that is everywhere, right? It's in Europe, in North America, and of course in Chile. And affecting Chile, PRV, different varieties PRV are affecting the 3 species farmed in Chile: coho, salmon, trout, and Atlantic. And in all those cases, it's becoming a concern, because at the end, when you have a viral disease that is kind of silent and it's not acute, it's chronic, you need to learn to live with it. So at the end, the immune system try to control these viral diseases and these agents. And at the end, when you find spots, could be the result of this reaction or defense of the body. So basically, what NOFIMA has shown is that one of the ways to prevent that is to use adequate levels of omega-3s, EPA DHA, basically. So that's a good approach that's documented, and maybe Brett can highlight more details than myself. But basically, being in practical terms, yes, you're right. Those numbers we have seen figures of 25 and even more in some groups. And the main concern is when you're sending whole fish, because when you fillet or you do portions, you can still sometimes remove and observe and remove those spots and eliminate. Even though you have a loss in the process, which could be substantial, you can get rid of it. The problem is when you send a whole fish, fresh fish to overseas, and then the customer opens the box and split the fish and finds the melanin spots everywhere in the fillet. That's not nice, and nothing you can do. Probably the fish will be just rejected. So the economic consequences in your branding, in your image is huge. So that's something that definitely need to look at. Now, the coincidental thing, and with this I finish, is that in the last 10 years, Glenn showed how the steady decrease in the omega-3 contents in the diets and, of course, in the flesh of the fish has become, and that's the result of over demand versus a fixed or declining supply of fish oil and fish meal. I mean, this industry has been based on those fantastic raw materials in the last 30 years, and without them, it would have been impossible to have what we have today worldwide as a salmon industry. They have been a tremendous asset to the industry. Yes, the problem is that they just cannot produce enough. So 10 years ago, we just broke that demand-supply curve, and fortunately today we have other options to compensate that difference. So the hope is that we will be able to come up to the 10 years ago levels of inclusion and probably overcome these issues.
Very comprehensive answer. Thank you very much for that. And I think you're quite right. I mean, what's possible today in terms of nutrition is completely different to what was possible 5 years, even 3 years ago. We have more tools at our disposal today, so therefore we're able to do things that we weren't able to do before, which I think is fantastic. And I've seen, the topic of melanin spots obviously is very specific, but more broadly, fish quality can be impacted positively by paying attention to omega nutrition in terms of color, in terms of yield as well, and clearly, obviously, in terms of fortifying the level of omega-3 in the fillet for consumers, which is something we need to think about too. But then there's some more questions in here. I want to bring you back in, Jorge, with a question on the tip of the tongue for many of the listeners today, which will be around the return on investment for Optimum Nutrition. If you're going to go back to higher levels of omega nutrition in the diets, there's going to be a price tag to that. So what's your message to farmers, and how should they think about this? And also, how do you think Veramaris can help?
Yes, as Fred mentioned, increasing the EPA and DHA levels and also arachidonic with Veramaris, and a proper balance of these essential omega-3 fatty acids has an impact under challenging conditions on fish performance, fish flesh quality, and fish health as well. So besides Veramaris as a sort of natural marine algae rich in EPA DHA, this does allow farmers to increase the level on flesh and on the feed, and also secure a minimum level of EPA while reducing the dependency of forage fish, and also open doors for those markets that retailers are demanding sustainability sources or less dependency of the ocean to protect the ocean. So finally, the ROI, or the return of their investment, can be obtained through better fish performance, less downgrade, like you already mentioned, like melanosis indices, and also better sales price. If you are able to fulfill customer expectation and retailer expectation, you also are going to get a better sales price, and also penetration of new markets, or a combination of all of them, where Veramaris can offer a non-marine product with EPA and can fulfill through Optimum Omega nutrition benefits.
Very good. And of course, these are all things that can be modeled, in terms of quantifying what the incremental cost of fortifying the feed with higher omega-3 levels can be, versus what the benefits can be both in the production phase through the things you mentioned, and then also the in-market benefits that we can get as well. That's a model that we've been talking to customers about over the past couple of years. So I think we are approaching the top of the hour, and we've answered quite a few of these questions, and we'll follow up on the ones that we haven't been able to address afterwards. So I just want to thank the panel for their contributions today. That's been a great conversation based upon a really good presentation from Brett, and that leads me to try to sum up a little bit. I think overall we're entering a really exciting new era where alternative ingredients like algal oil are now available at scale, and they're affordable, too. So this creates huge opportunities for salmon farmers to take back control of their omega nutrition and to decide what level they actually want to have in their feed, depending upon what their farming circumstances are and what challenges that they're trying to address within production. And the benefit of that will be seen very clearly in terms of fish health, fish welfare, and also productivity too. These are things that are all documented as benefits of optimum omega nutrition. So I hope that the webinar today has been informative and interesting for people who have logged in, and I encourage you all to continue the conversation with our regional representatives. Please join me in saying thank you to the panelists for today, and look out for the invitations to future Veramaris webinars, which will be coming soon. And in the meantime, I want to wish you all a very safe and happy holidays. Thank you, everybody.
Thank you.
Thank you very much.
Thank you very much. Bye.